start from where I have left. Okay. So before that, I wanted you guys to understand that how it's inevitable process because of the natural wear and tear of our cellular machinery. It will happen sooner or later. All we can do with our lifestyle, with our with our way of living, and by helping our body, all we can do is to delay that inevitable phenomena. We might be able to delay that phenomena for a longer period of time, and then for a longer period of time, then for a longer period of time. And the more efficient we able to be, in using our resources, the the less chance of we will be having developing cancer in earlier stages. So if you ask me, in today's perspective of the world, developing cancer at the age of 80, 90, and 100, it make all the sense to me, because your machinery is getting older, the control of the things is getting difficult, and it make all the sense that sooner or later those people will going to lose the control on their cellular machinery. But having said that thing, developing cancer at the age of 25, at the age of 35, 40 means that you have misused the resources you are carrying. And last but not the least, if somebody is unfortunate enough and he received the erroneous genetic code, he receives the genetic code with mutations, and that person might not be able to avoid it, because these errors will going to push the things, or will going to push the cancer sooner than later. But what happens biologically at that time? It's about the cell G.
So this is just something, I don't know whether it's true or not, but पहले ज़माने में लोग used to have really long lifespans, like hundreds of years wala. So why didn't cancer appear then?
Of course, there are. I can answer this thing. First thing is that the average age of human life is increased over the period of time. But that understanding does not fit with our knowledge of prehistoric people, prehistoric people. जो आपने सुना हुआ ना, जो scriptures में पढ़ा हुआ है, hundreds of years of life at that time. जो मैं आपको बता रहा हूँ, that does not fit with those things. If the data we are having as a researcher for the last few thousand years, recorded history में, the human life is like an ups and down for many thousand years. But for the last couple of thousand years, when the recorded history was more established, we only observed a gradual increase in the average life of human beings. So now we are having a stable increase in human lives. How I can justify the presence of those hundreds of years of life before that? That is only applicable, that is only, it can only be explained with the philosophy I have explained you in the earlier class. That if you are using your resources in the best possible way, if you are giving the body the absolute things the body is required, if you are excreting the things well, if you are metabolizing the components well, if you are living and breathing in a safe environment, that there is a high chance that you will not be able to develop the same things in earlier years. This is the same thing I told on the other day, that sooner or later, I mean, there are, from few thousand years from now, I believe that people will gradually started living lives of those hundreds of years. If you give only the nutrient which is required for the metabolism to human, if you take all the unwanted material from the body of humans, if you allow that humans to live without any stress, if you allow that humans to breathe with the pure oxygen, if you allow that human to to execute their metabolic chemical reaction with wonderful efficiency, you can guess what will happen. The extreme efficiency of those things will give us a long expectancy of life. So that question can be addressed using the same thought which I have shared earlier. So one thing the cancer cell is doing, and that thing is that it is the independent of the growth signal. Usually a cell receives a signal that when it is required to divide. Usually cell do not go and proceed for cell division without that signal. This is called, this signal might be coming from the growth hormone or from something like that. If a cell ignores the presence of the growth hormone signal and start dividing without even having a growth signal out there, that means the cell is developing a characteristics of cancer. And these things can be triggered from the internal reasons as well. And those internal reasons are called as oncogenes, cancer-causing genes. Some cancer-causing genes push the cells to go for the cell division even in the absence of a growth factor or a growth signal. So without the growth signal, the cell chooses to go for the cell division. The second thing is that once the cell started undergoing cell division or the cell cycle, after a reasonable time, when the cell and the organism believes that the cell division is no longer required, the organism generates stop signals and controllers and breaks. Those stop signals help the cells to feel that the cell division is no longer required. And the cell cycle gradually decreases and cells stop cell division. What happened if the cell started ignoring those stop signals? If the cell ignores the stop signal or the mechanism in which the stop signal is communicated to the cell is gone, so what will happen? You can guess. The cell will keep on cell dividing, keep on dividing, keep on dividing. And that will going to produce a mass of cells, irregular mass of cells, and that's what we call as tumor. And usually these things are related to the genes and the proteins which are controlling the cell cycle, which are controlling the errors. So in general, if you understand the biological aspect of cancer, believe that if a policeman or the judiciary or the law enforcement agencies of the cells, their job is to keep the law and order situation within the cell. If they gone corrupt, if they are not following the standard operating procedures, the cell will not going to control itself, and that will start dividing and producing large number of cells and a mass and what we call as tumor. I hope that you explain the biological aspects of tumor. There are a lot of characteristics we can attach with the cells. For example, अहमद ने हाथ खड़ा किया जी. अहमद, आप क्वेश्चन करें, फिर मैं ये फिगर एक्सप्लेन करता हूँ.
अस्सलाम वालेकुम सर.
वालेकुम अस्सलाम.
सर क्वेश्चन तो नहीं था, लेकिन मैंने एक ना चैट हेड के अंदर आप एक बात लिखी है सर, वो जरा प्लीज उस पे गौर कर लीजिए.
आप रीड करते हैं, अगर आपकी मेहरबानी हो कि चैट हेड ने लोग आईडी लिख रहे हैं मेरे कहे बगैर भी, तो फिर मुझे डिस्ट्रैक्ट, एंड आई फाउंड के आईडी वो वाला क्वेश्चन का मैसेज आया. ओके, आप रीड करें तो हम सब सुनेंगे यार इतने मजे से. अच्छा. Oh, you can ask, you can told me these things later on, yeah. Even they can told me, okay. It was a request about the two students who were not there. Okay, thank you very much. Okay, I can proceed further with the things. Please let me know about these things after the class. Okay, let me explain the figure. Okay. So there are a lot of characteristics which we can attach with the cancer, and technically they are called as hallmarks of cancer. It's a very famous term. Remember, it's a very, very famous term called hallmarks of cancer. People use their terminology to talk about the cancer. So what the cancer cell is doing, every single term belongs to the same thing which I was talking. Think enabling replicative immortality. The cells started feeling it will go on for the cell division for the rest of its life. This is called replicating immortality. Feels I need to divide, then I need to divide, then I need to divide. There is no stopping for the cells. They become immortal in the cell divisions. Tumor promoting inflammation. The situation at the site where the cells are dividing, remember, cells are not able to divide if they are not going to receive supportive environment. If they will not going to receive supportive environment and conditions, and tumor supportive environment is there, that means cell divisions is going on, and also cells started feeling to have a cell division supported environment. And whatever the genomic instability is there, and lot of more mutations are out there. This is a very interesting point. I wish I should be able to teach you about this thing. Once we have a cell with a nucleus, the cell feels when it is required to go for the cell division. There is a certain type of things which should be there before the cell proceed for the cell division. Okay, is mahol ko samjhe. The first thing, the cell feels that there is a need for the cell division, and that need is actually supplied with the help of growth signal. Growth signal is a signal that there is a need for the cell division. Is there are enough nutrition out there, enough food is out there? That's the next need. And after that, if everything is set up, the cells do check the internal genetical system as well, that everything is fine. It can proceed for the next phase of cell cycle, which is called as synthesis phase, where the cells is required to execute the DNA duplication process. And the DNA duplication process requires the presence of all the nutrients and ingredients out there. If all the things are fine there, the cell choose to and commit the cell division. Remember, it is very serious for every cell to commit the process of cell division. Very difficult. You know why? Because once the cell decided or committed the cell division, there is no going back. You cannot reverse the cell division. Duplication हो रही है, थोड़ा सा DNA बना लिया है, बाकी बाद में बना लेंगे. It is not possible. Once you started the cell division, that means the cell now must have to complete this process after that. Cells check every time itself along. So after checking everything, cells proceed with the cell division. And if something is not nice out there, something is not fine. Something is not fine means you have to create all the DNA once again. All the DNA, all the genetic material, all together once again during the cell division. And in that time, there is a chance of putting the wrong base pairs during the process of replication. So what will happen? There is a process called DNA repair mechanism, which is activated at that time. It started picking that what is wrong out there. Is there any mutation during the process of replication? If there is a base pair is placed on the DNA, if there is a trouble during the process of replication, and it fixes all those troubles at that time. Make sure that everything is nice, fine, and clean, and DNA is replicated perfectly, and then allow the cells to move to the next phase of the cell cycle. So it's a very complicated process in which the cell decides to proceed with the cell division. Now, what happens in a cancer situation? A cell usually ignores a lot of these things. For example, think about deregulated cellular energetics. What that means? The cell feels that I have enough amount of glucose or energy creating molecules out there which are enough to execute the process of cell division. You know, imagine if the cell is not, if the nucleus is not there, the cell will not be able to do the normal things. Jab cell division hoti hai na, I can tell you, for some period of time, cells do not exist. We are dead at that time. Because there is no nucleus, there is no normal process, no normal function of nucleus. If the nucleus is not there, imagine nobody is there to control the functions of life. ये एक इंतहाई खतरनाक जंप होती है जिसके अंदर डेथ हो सकती है सेल की. So the cell is required to check everything is there before it enters. ये डाइव है ना, एक डाइव है समंदर में, उससे पहले चेक करते हैं कि everything is fine. So if the nutrients are not there, the cells holds itself, does not go for the cell division. But if the cell is cancerous, so it can avoid that thing as well. It can change its energetics, it can metabolic pathways, it can utilize its proteins and other resources to produce energy and go for the cell division. Usually if the mutation is there, cells feel I need to stop for the cell division. But in case of cancer situation, even with the mutations, cells pushes itself to go for the cell division. So there is not a push for the cells to go for the cell division in normal conditions, but in cancer situation, the cells feel that there is a pressure to proceed for the cell division. And under that pressure conditions, when cells proceed for the cell divisions, it develops mutations. And you can guess what will happen. The number of mutations will be more in the next generation. The chances and the probability of developing mutations will be more, because the cells does not proceeding for the cell division in a normal way. It is proceeding the cell division in a push. Like, I need to check for the food, the cell is not checking for the food, go for the cell division. I need to check for the mutation, the cell is not checking for the mutation, going for the cell divisions. So even all of them or one of them can be a reason to push the cells to go for the unwanted cell divisions. And as a result, the new cells, they will be smaller and they will be rapidly dividing. A cell is supposed to grow and then divide. But in case of cancer, they just created and then they start dividing once again. Created and start dividing once again. That makes no sense, because they are not supposed to do, they are supposed to grow and appear and become something which they should be. They might become like an epithelial cells, but they're not becoming epithelial cells, and they are just like dividing. All of their focus is in the cell division. Thank you very much. All of their focus is on the cell division only. And that's very pressure or unkind way of going on with the cell division. And you can sense what will happen. It accumulates lots of mutation in the genome, and that disrupt and disturb the normal functions of the cells, and that can cause the genomic instability. These mutations, the cells will become worse and worse, and you never know what type of mutations it is carrying. So all these hallmarks of cancer are characteristics of the cancer cells. And I have explained these things, but just for your understanding, I am just going through that during the cell cycle, there are a certain checkpoints present between the two cell cycle states. The one is between the G1 and S. This is called as G1S checkpoint. The cell thinks and feel that it is required to enter into the DNA replication mode or not. It is required to proceed in the DNA replication mode or not. Whether everything is fine, everything is nice or not. Everything is fine and nice, it goes into the DNA replication mode. I can tell you from here to here, there is no cell. There is no cell. Right? I mean, there is no nucleus. There is no cell at that time. It's all about somebody, a genetic material is opening up, is replicating or undergoing mitosis. So all the things which is required for a cell to survive at that time should be there. These are the things which the gates which actually make sure that everything is there are called as checkpoints. So there are cell cycle checkpoints which make sure the cell should proceed to from G1 to S or not. They are called as G1S checkpoints. There are other checkpoint, very famous one, which is present between G2 and M before entering into the mitosis. This is the final step. This is like the final checkpoint. Whether I should allow that cell to go for the actual cell division, which is called mitosis. It only allows the cells to go for this phase when the DNA is replicated perfectly. The DNA is replicated perfectly. If the DNA is not replicated perfectly, the DNA machinery will be able to pick this thing up and it will stop the cell division before the mitosis, and the bad cell will never be able to go for the cell division after that. But what if the machinery which is responsible for analyzing all these things, if that machinery is not there, if that machinery is not working fine, it will allow the cells to proceed without any check and balance. And that errors will be passed on to the next generation. And that is a very, very serious state. Okay, so the cell cycle should be triggered with the help of the growth factor. There should be somebody who is telling the cells to proceed, and through the signaling pathway, it should the message should be transferred to the genes and to the cell cycle. But what if the cells is ignoring the growth factor? Even in the absence of the growth factor, the cell will start pushing the cell cycle to go further, and that is a very unfortunate situation. Also, there are tumor suppressors. Their job is to control the cell division. They regulate the cell divisions. For example, a tumor suppressor is supposed to produce a protein which acts as a brakes, and allows the cells to divide with a certain speed and rate. But what if there is a problem in the tumor suppressors, and that can the functional protein is no longer be there, and there is nobody who is controlling the cells for their proliferation, and we have lots of cells and a collection of large number of cells which we call as the tumor. Okay, so that's it. It can also be a reason. Okay, so this is all I have talked about. You will see it's easy for you. I have for the last 2 lectures I was talking on these things. Some cancers, they are supposed to stay at one place. They are called as benign tumors or benign cancers, but the others, they are supposed to change their locality as well. This is called malignant tumor, or and the process of spreading the cancer cell from one place to another is called as metastasis. So tumors can be classified into different types, depending on their sizes, depending on their structure, depending on the presence and absence of certain receptors. And usually there is a diversity in the cancer cells. I believe that you might be able to feel that if we have 100,000 cells in a tumor, they are not of the same type. They are like, they are made up of different types of cells, different combination of cells. And all these different combinations arises from some original cells. And why in the tumor we have large number of different type of cells? You can guess that is because of the ongoing mutations. In one case, I just want to give you one example and we will proceed further. There is a very famous gene called BRCA. सुना होगा, पागल हो गए होंगे सुन के इसका नाम. मैं आपको बताता हूँ ये काम कैसे करता है. BRCA1 सुना है आप लोगों ने? जाग रहे हैं?
Yes, sir.
Yes, sir.
और तुम्हारी मेहरबानी मेरे नाल रहना. I will going to teach you some. ये को मैं बहुत अच्छा साइंसदान किस्म का टीचर तो नहीं हूँ, but these are very important things. Okay, I'm trying to. BRCA1 काम कैसे करता है? ये सबसे फेमस है. अगर आप किससे पूछे ना कैंसर में, ब्रेस्ट कैंसर में रिसर्च करते हैं तो पहला नाम वो लेगा ना, वो P53 का लेगा और दूसरा नाम जो है वो BRCA1 का लेगा. Let me tell you the mechanism कि ये काम कैसे करता है. BRCA1 जो है ना ये DNA, it's a DNA damage repair pathway protein. यानी कि DNA damage repair pathway का एक प्रोटीन है. And what that means, if there is an error in the mutation in the DNA, so BRCA1 will be there, and that BRCA1 will going to fix the error. जो replication के दौरान या mutation के दौरान गड़बड़ हो जाती है, तो ये उसको fix करता है. Now think if the BRCA1 is not there, तो DNA errors को fix करने वाला कोई नहीं होगा. What that happens, the error will be passed on to the next generation. वो कौन सा error होगा? Who knows? The next cell will going to divide as well. It contains mutated BRCA1. That means when the cell divides, it produces undergo replication, develop some mutations, and that mutation will be passed on to the next generation. Nobody is there to stop the mutations as well, to fix the mutation as well. Now tell me, the new cells of the 3rd generation and the 4th generation and the 5th generation, they will going to develop mutations randomly on different places. Nobody can categorize कि ये mutation कहाँ पे होंगी. Nobody can feel, can sense कि ये mutation किधर होंगी. These mutations will be random on any place on the genome. It will going to create lots of different types of cancer cells. Some of them might be benign, some of them might not be. Some of them are less proliferative, some of them might be very much proliferative. So we will have a heterogeneous situation within the tumor. That is why it is very difficult to rationalize the tumor and to define something to fix the cells. Okay. So these are the properties. There are certain basic things you should know about. I should teach you these basic things, but unfortunately, I'm teaching the other things. The carcinomas means the cancer of the epithelial cells. इसीलिए breast cancer को breast carcinoma कहते हैं, because it's the cancer of the epithelial cells of the breast lining. So इसलिए इसको breast carcinoma. अगर किसी कैंसर का नाम सार्कोमास लिखा हो तो इसका मतलब है कि ये कनेक्टिव टिश्यू का कैंसर है. आपको याद होगा, आपने बोन का कैंसर जो है, टिश्यू का कैंसर जो है, सार्कोमास कहलाता है. If there is a leukemias, lymphomas लिखा हो, this is the cancer of the tumor referred to as the leukotumors, from blood cells and from the white blood cells, T cells, beta cells के से जुड़ा हुआ जो कैंसर होता है उसको, जो immune system का कैंसर होता है उसको बोलते हैं lymphomas. Okay, so before I finish, there is I mean we have 2 minutes, and let me see that I able to click this one thing or not. You have to come back again, okay, after the after finishing that meeting, so I can complete my conversation with you guys. Okay, any question? And let me complete the remaining part of this lecture. Okay, I am recording my session. I believe Abir ji, Abir आपने कुछ पूछना है? आपने hand raise किया, शाबाश.
Sir, how does benign tumor turn into a malignant tumor?
It's a
Like
very
I
good.
heard that it can be induced. So how does that happen?
It can be induced नहीं. I mean ये बड़ी मजे की स्टोरी है. मैंने उधार रखी हुई है अपने ऊपर. मैं आपको बड़ी मुश्किल से कम पढ़ा रहा हूँ. I'm sorry to tell you about cancer. It's so difficult for me to stop myself. But there are a lot of factors which I should talk on. Okay, how a benign tumor is actually become so bad that it started changing its place and it's moving to the other places and causing cancer. हाय ओके मैं सुनाता हूँ आपको कुछ करता हूँ इसके बारे में. ओके नहीं तो मैं फिर कभी सुनाऊंगा नेक्स्ट क्लास में. when the cells choose to grow, मैंने आपका सवाल पकड़ लिया अपने पास. ओके डोंट वरी. normal cells when they grow after a certain period of time they stop growing. उसकी बेशुमार रीजंस होती हैं. they should supposed to stop when they started touching each other. It is called contact inhibition. Cancer cells, in case of cancer cells, the contact inhibition factor was also compromised. And they even grow when they are like, they touch each other, they even grow after that and after that. Now, there are a lot of bad things going on within the cells. So they just kept on growing, and it is not only growing, they're developing newer type of mutations, so becoming different and different and different. Okay, the important thing is that whether a mutation will going to help a cancer cells to live or will not going to help the cancer cells to live. We don't know. We don't know the answer. Because the mutation is a random situation. If we have like 100 different types of mutations during the cell division, the likelihood of that the 99 mutations out of it are not supporting the cancer phenotype. But what if the one mutation which helps the cancer cells to live in harsh conditions, it allows the cancer cells, enables the cancer cells to fight and to survive. And that one mutation will going to help that one cell to live. The rest of the 99 cells will be gone, but that one bad cell will going to live and it will going to produce more cells. So this is called selection of the worst, and then selection of the worst, and then selection of the worst. Eventually we will be having a very bad type of cells, which we call as cancers. A benign or localized tumor is the one which actually stays at one place. Frankly speaking, benign tumor is not the reason of death in most cases, because benign tumor or the tumor at one place is not a threat for anybody. It's like a living cells, it's like human cells. It's not causing a lot of trouble, not attracting any immune cells, not putting anybody into trouble, apart from the fact that it is actually creating a mass at some place. If that some place is important, like brain, then it's a threat. If that some place is not very much important for the functions, overall function of the body, that might not be a threat. If benign tumor is present in the breast, it does not going to affect anything as long as it is benign. But the real killer is the ability of the cells to move and to change their place. That's what we call as metastasis. You can see the dark cells, they are the cells, and their ability to change and move their places. So this is supposed to be the this is called carcinoma or epithelial cells cancer, and it is a malignant version of the cancer. And in this case, what happening? The cells are, you can see the cells are leaving off their places from their collection, from where, from their source, it is actually moving to the other organs. And that is the situation when the cells, they choose to start or affecting the normal functions of life. And that is how they're able to kill the person. Everything starts from the place from where cancer arises. In case of breast cancer or any sort of carcinoma, there are cells, different types of cells which are creating the lining of the cells. If some of the cells, the red ones, look at the red ones, if some of the cells, they started having a cancerous phenotype, means they started growing, replicating and proliferating more than normal, they will going to outnumber the local cells. They will going to outnumber the local cells. And their numbers increases and increases. But as long as it is staying here, it is a sort of breast benign tumor. You can consider it as benign as long as, although it is increasing in size. But once the cells having the size of the tumor is increased and increased and increased. The cells require more food. The internal cells require supplies. It requires glucose. It requires oxygen. So the system, for their survival, they need to supply or create some channels in order to supply food and nutrients to the innermost cells of the tumor. This is what we call as angiogenesis, producing more blood capillaries. Producing more blood capillaries will going to give more blood and oxygen to the internal cells, and they will going to flourish as well. Abhi tak ko khadre ki baat nahi hai. But the next thing is that when the cells choose to leave their place, this is what we call as malignant carcinoma or metastasis. It does not happen in one stage. It actually happens in multiple stages. It happens in multiple stages. At one place, the cells choose to leave their locality. I'm not telling you everything about that. So this is not a major subject of cancer biology. I just need to touch those things. So maybe, but I can't stop myself. I might use extra time to explain this phenomena in detail, that how a single cell is able to move from one place to another. So the cells leave their place. They're going to survive in the blood. Every single step is like a hell for the cancer cell. It is not possible for any cell to leave their place. Remember, epithelial cells are only able to live and survive among the epithelial cells. They will never be able to leave their place until unless they change their phenotype. They will become like different. They will become like mesenchyma cells. Only then they will be able to live. And this is what we call as epithelial to mesenchyma transformation, EMT. So without EMT, the cells are not able to live outside their locality. That is required. And that helps the cells to leave their place, and that helps the cells to live in the blood cells. And even in the blood cells, they will be dead because of the actions, because of the environment and the system out there. And they are required to exist. Most of them will be killed in the pathway, but some of them, they develop enough mutations and enough phenotype that they're able to survive out there. When they found a suitable destination where they can interact with the local cells, they found and develop their interactions, and then eventually they invade the target cells or the target organ and start increasing and growing their numbers out there. So you can see the source, the starting point, and then the destination point. This might be the breast from where it is arising, and this might be the brain, this might be the bones from where the cell started creating a secondary tumor. Now, if you want to kill those cells, you need to remove the cells from all these places. If you know a mass of cell is out there, you can remove it by surgery. Doctors try to use the surgery to have like to remove a maximum chunk, including the normal cells as well from this place, so that there is no chance that there is not any single cancer cell should be living out there. But they might remove this thing, but they might left some of the cells, in the veins, in the surrounding areas, and those cells will be able to produce the tumor once again. So there is no definite way to remove all the cancer cells.
Sir?
Yeah. Gigi.
Sir, is this what we call fourth stage of cancer ke puri body mein spread?
Yeah, staging of cancer means that it's somehow a yardstick to measure how bad that cancer is. And of course, it includes a lot of factors, including the heterogeneity. If you remember the concept of heterogeneity, that how many different types of cells are out there, whether the cells are staying, they are leaving, or they are metastasizing. All these things together define the stage of the cancer. There is a tumor grade term as well. You should know. Tumor grade is a different thing, and then the stages, cancer stages, and of course they depend on these things. Gee, you guys are pushing me to places where I don't want to talk. Gee. Shabash.
So if the tumor is staying at one place, it's not moving to other place, as you said. There's a category of tumors. So why do we need to remove the tumor then?
Yeah, we don't need to remove the tumor at that time if it is not spreading, but we need to remove the tumor because we don't know whether they will going to stay as a benign tumor or not.
Okay, I mean, if they do have the tendency to spread.
all the type of metastatic tumors, they come from the benign tumor. किसी ज़माने में वो तो वही होंगे ना. So that is why it's important to remove them. So you should not give or leave any chance of complication. Yeah.
Sir,
जी जी शाबाश शाबाश.
if someone is genetically, inclined to have cancer and all, so why can't we just induce something in the benign tumor so that we can detect it afterwards, like a fluorescent gene or something? We just see the cells and extract only those.
But we need to know the practical implications of what you are speaking as well. Imagine there is no injection which you can inject into a person's body and it will take the gene right inside the nucleus of the tumor cells only. So we need to think that how we will be able to put the things selectively in the tumor cells only. Okay, so you need to think what are the ways that we can inject or insert the genes right into the genome of a cell, and how we can distinguish that the fluorescent gene which we are inserting, it will only going to enter into the benign cells. It is not going to enter into the normal cells. ठीक है? बस एक बातें हैं.
Yes, sir. That's a tricky situation.